Hello, my name is Cana Quave, and I'm a PhD student in the DeMonte Lab at the University of Texas Health Science Center at Houston. Thank you for viewing my poster. Opioid use disorder and associated overdose deaths have drastically increased in prevalence in recent years, despite the obvious risks associated with opioid drug misuse. Studies suggest that deficits in risk-related decision-making in opioid users occur alongside alterations in prefrontal cortex activity. We hypothesize that repeated opioid administration blunts the prefrontal cortex response to threats, thereby biasing behavior toward reward seeking and away from threat avoidance during motivational conflict. To test this hypothesis, we implemented single unit recordings from the prelimbic subregion of the medial prefrontal cortex, or PL, of rats while the animals were exposed to an approach avoidance conflict task. For this, we used a modified conditioned place preference protocol. Adult male Long Evans rats were conditioned every other day with either morphine or saline, and the side of the apparatus preferred least at baseline. Conditioning occurred over a 10-day period for a total of 5 drug pairings and 5 saline pairings. Immediately following preference testing on day 13, we placed a predator-associated stimulus, cat odor, in the previously drug-paired side to create an approach avoidance conflict for the animals. We also recorded the spontaneous activity of PL neurons before and after the first drug administration on day 2, before and after the final drug administration on day 10, as well as during the entirety of the preference and conflict test on day 13. In Figure 1, we showed that morphine-treated animals demonstrated a locomotor sensitization effect, as evidenced by an increase in the distance traveled in the drug-paired side across conditioning days. During the preference test on day 13, as shown in Figure 2, morphine-treated rats displayed a clear preference for the drug-paired side of the apparatus, increasing the amount of time spent in that side as compared to baseline. This preference was also clear when we looked at raw time spent in either side of the apparatus during the preference test, with morphine-treated rats spending greater amounts of time in the drug-paired side than in the neutral side. During the conflict test, which was performed immediately after the preference test on day 13, saline-treated animals avoided the side of the chamber in which the cat odor stimulus was presented. In contrast, morphine-treated animals continued to enter the previously drug-paired side despite the presence of cat odor, suggesting that morphine conditioning induces an increase in risk-taking behavior. Given the PL's role in processing both rewarding and aversive stimuli, we next investigated whether morphine might be affecting PL neuronal activity throughout the course of the experiment. In Figure 4, we showed that after acute morphine administration on the first drug conditioning day, a significant number of PL neurons showed a suppression of firing rates. In Panel B, we can see representative units from the saline and morphine groups, with the representative cell from the morphine-treated animal showing a clear reduction in the number of spikes recorded following acute drug administration. However, as we can see in Figure 5b, the broad inhibition of PL neurons observed after acute morphine administration was clearly absent on day 10 after the fifth and final drug administration, suggesting that PL neurons become less sensitive to the effects of morphine following repeated exposure to the drug. Next, we sought to identify whether PL responds differently during the approach avoidance conflict test after morphine conditioning. In Figure 6, we compared spontaneous firing rates of PL cells when animals entered the side of the apparatus containing cat odor to those of the same cells when animals entered the neutral side during the conflict test. In saline-treated animals, cat odor side entries were associated with an increase in firing rates of a significant number of cells. However, in morphine-treated animals, this pronounced ratio of excitatory neuronal threat responses was not observed. Based on our findings, we conclude that morphine conditioning leads to a reward-related behaviors commonly recognized to be characteristic of addictive processes, including locomotor sensitization and the expression of contextual reward memory. With repeated morphine administration, PL neurons undergo a physiological adaptation that renders them less sensitive to the effects of morphine over time. Additionally, as we hypothesized, morphine conditioning biases rats' behavior toward reward seeking when a considerable risk is present. Our most intriguing finding is that during the conflict, firing rates of PL neurons during threat approach shift from predominantly excitatory responses in the saline group to equal numbers of inhibitory and excitatory responses in the morphine group. We conclude that this loss of excitatory signaling of threat in PL might underlie risk-taking induced by repeated opioid use. Thank you for coming to my poster. If you have any questions, feel free to contact me. Thank you and take care.